In silico screening of traditional medicinal plants and compounds found in Camarines Sur and Albay as potential SARS-CoV-2 inhibitors. The COVID-19 pandemic has been the talk of the year 2020. The spread of the SARS-CoV-2 virus will continually be a major concern in the preceding years without proper medical intervention.
Aggressive implementations of suppression strategies such as case identification, quarantine and isolation, contact tracing, and social distancing have already been implemented. However, this does not ensure that the spread of the virus can be prevented, as transmission will quickly rebound if interventions are relaxed. Thus, COVID-19 vaccines that can be used globally is therefore the ultimate solution for ending the global pandemic.
The problem, however, lies in the global distribution of vaccines. In the Philippines, persisting issues are clearance concerns and the prioritization of high-income countries. Vaccines will not be accessible anytime soon in far-flung areas due to the limited resources and aligned priorities.
For developing countries like the Philippines, achieving mass immunization is not expected until 2023, a report published by the Economist Intelligence Unit or the EIU has inferred.
Furthermore, vaccination is only beneficial for disease prevention and added protection from virus exposure. Currently infected individuals would not be allowed to administer vaccines. Non-availability of drugs will prolong their recovery from these symptoms, which may result in untimely deaths in adverse cases.
For the meantime, the current therapeutic approaches are the solutions used in previous pandemics. In the study of Wang and colleagues 2020, the remdesivir was identified early as a promising therapeutic candidate for COVID-19 because of its ability to inhibit SARS-CoV-2 in vitro and in vivo. Moreover, remdesivir also showed a significant antiviral activity against a diverse panel of notable RNA viruses, such as Ebola virus, Marburg, MERS-CoV, SARS-CoV, respiratory syncytial virus, Nipah virus, and Hendra virus.
Moreover, the urgent need for therapeutic agents has resulted in repurposing and redeployment of experimental agents and medications used for other diseases. These include hydroxychloroquine, an anti-malarial drug, lopinavir or ritonavir, a medication for the treatment and prevention of HIV or AIDS, sarilumab, tocilizumab, and convalescent plasma therapy.
Unfortunately, these existing solutions and treatments are not as optimal as they are expected to be. While remdesivir is the established compound proved to be the most promising in the antiviral drugs, it still is a commercially formulated and scrutinized compound, not found raw in natural sources. Furthermore, the unfavorable inventory status of other developed antiviral drugs makes it difficult for us to procure them, as these antivirals are foreign products and thus require a massive budget. Additionally, chloroquine, which was also a leading candidate against SARS-CoV infections, also had side effects based on numerous clinical trials done to test its efficacy. Studies found that not only did patients not do better if they got the drug, but they were also more likely to suffer from minor risk of macular retinopathy and in some cases, cardiomyopathy as indicated in the study of Easterbrook 1993.
Besides synthetic drugs, nature has also played a crucial role in the development of many medical drugs. Local healers have already been accustomed to using herbal medicines as the first treatment to any disease, as part of their culture and traditions. Influenced from the country's pre-colonial healthcare system formed by Babaylans or Balyans, albularios present a diverse extent of materia medica that have been used for medicine.
Moreover, almost half of the approved drugs in the past 30 years have been of natural origin. Almost 25 to 70,000 plants have already been evaluated for medicinal use. Some examples of plant-derived medicines are as follows. Nitisinone, which is derived from the natural product leptospirone or Callistemon citrinus, is used in the treatment of tyrosinemia. Galantamine, which is a natural alkaloid, is obtained from Galanthus nivalis and is used for the treatment of Alzheimer's. Lastly, apomorphine is a semi-synthetic compound derived from morphine. Papaver somniferum is utilized to treat Parkinson's disease.
Given the fragile medical response despite the existence of a variety of therapeutic approaches, several studies, mostly in the pharmaceutical industry, have been progressively targeting plants with the aim of identifying lead compounds, focusing principally on appropriate alternative antiviral drugs. Our study, which will include traditional medicinal plants and compounds found in Camarines Sur and Albay, acquired its objectives and inspiration from these existing studies about herbal plants and compounds as potential SARS-CoV-2 inhibitors.
Yunesi and Rizazade 2020 in their review suggested possible SARS-CoV-2 inhibitor plant species, Urtidica dayoka, Lin, common nettle or alingatong, Scutellera baikalensis, Georgi, Baikal skullcap or sidit, and Nicotania tabacum, Linn., tobacco or tobacco. Upon evaluation, the researchers have observed that these species contain plant lectins, which are carbohydrate-binding proteins that can act as immunologic receptors and defense proteins. So consequently, these findings suggest that plant lectins could be significant in finding an antiviral immune response on humans.
Interestingly, multiple lectins have been shown to neutralize a number of viruses, including HIV, making them attractive targets for the development of novel antiviral drugs. To further discuss the antiviral mechanism of plant lectins, let us see this example on the anti-HIV activity of lectins.
This model on the left shows a bird's eye view of the trimeric structure of envelope spike, a protein on the HIV surface that enables it to infect cells. As seen on the image at the right, this envelope glycoprotein consists of two subunits, including GP120 and GP41. The viral cell fusion process of HIV is initiated with the binding of subunit GP120 to the cell surface CD4 antigen, a glycoprotein found on the surface of immune cells. Now, due to the virus's heavily glycosylated nature, this entry event of the virus may be disturbed or blocked by agents that are strong enough to interact with the glycans associating the viral envelope proteins with host cell receptors, eventually preventing the establishment of the provirus. Moving on, another significant study by Keaka and colleagues 2020 also demonstrated the potential antiviral effects of plants and natural compounds found in plants. The researchers found that cycloponins A, B2, C, and D, and licorine found in species Lycoris radiata show anti-coronavirus activity at given concentrations. Not only this, but their study also evaluated plants that we usually see in our backyards such as echinacea, ginseng, garlic, and ginger, and later found out that they are effective plants known for their immunostimulatory and anti-infectious activities.
The plants mentioned that were inferred by the researchers to be SARS-CoV-2 or coronavirus inhibitors were of international origin. Some may be found in the Philippines, but not as accessible to be consumed by the localities. However, the Materia Medica in our country can already give us an overview of compounds and plants we can explore to find potential SARS-CoV-2 inhibitors.
Furthermore, the utilization of Materia Medica essentially widens the scope of availability of drugs and highlights the significance of local herbal medicines in an important drug-dependent society. It is because so far, research done in this field of study increments the credibility of using these plants for medical utilization, as said in the study of Lazarte 2020.
Folk healers in Albay and Camarines Sur were investigated to tally usual plants they use in their healing. Folk healers in Partido District, Camarines Sur, used plants of which some are in the families Anonaceae, Asteraceae, Ruticeae, Poaceae, Euphorbiaceae, and Lamiaceae. Examples of local plants under these families are as follows.
Dorosan et al. Found out that 44 medicinal plant species are being used by health practitioners in the province of Albay, specifically the barangay health workers. Sambong, luya, avocado, buyo, and artemisa are some of the widely used Materia Medica found in the locale. It was also found out that among the medicinal plants identified, Cucus nucifera or coconut or nyog, Manihot esculenta or cassava or balingoy, Psidium guava or guava or bayawas, Gliricidia sepium or Kunth X walp or quickstick or more commonly known as Gliricidia or cacahuate, Euphorbia hirta or garden spurge or gatas-gatas, Philanthus niruri or stonebreaker or turutolikot, Ficus pseudopalma blanco or the Philippine fig or lubi-lubi, and Blumea balsamifera or camphor or lakad-bulan contain the highest importance value. The said plants in the summarized tabulations of Serio and Dorosan et al. Can give us a brief overview and insight into what plants we can examine in the completion of our research goals.
In addition to these lists of plants, we have also tabulated a tentative list of plants and their compounds that are potentially known to be SARS-CoV-2 inhibitors. The list is as follows.
We have three filtering mechanisms in creating the tentative list of plants with their respective compounds. We have used three different mechanisms to fully exhaust all possible compounds in the wide array of established resources. The three methods also involve the use of these two databases, namely UPMTK-LBH and Dr. Dukes. The first method includes creating a list of the literature-suggested antiviral compounds, searching a compound from this list in Dr. Dukes where the said database suggests plants with that particular compound, and manual search and cross-referencing of the plant in Camarines Sur and Albai's Materia Medica in UPM TKLDH.
Another method we used is the referencing to UPM-TK-LDH's Materia Medica of Albay and Camarines Sur for a plant, searching for the plant in Dr. Jukes to know their compound composition, and cross-referencing with their initial list of compounds from related literature. The final filtering mechanism we used includes the pinpointing of a plant from UPM-TK-LDH's Materia Medica of Camarines Sur and Albay, checking of compound composition in Dr. Jukes, and cross-referencing of compounds with the studies that suggest potential antivirality in the internet.
To accomplish our methodology, we have taken and modified methodologies of previous similar researches. The evidence of use of in silico filtering of these compounds was emphasized in Agrawal's study in 2020. Agrawal and colleagues conducted an in silico study through molecular docking of 10 herbal molecules investigated to predict their antiviral efficacy in the active site of the target enzyme. Software applications such as ChemSketch 2015 freeware was used to prepare the 3D structures of the said molecules, and Autodoc 4.2 and Discovery Studio 4.5 to conduct the actual molecular docking. Some examples of the interactions created by the researchers are as shown.
Another notable study by Zhang et al. Also indicated the use of in silico filtering as their main mechanism in the determination of potential SARS-CoV-2 inhibitors in plants. Their study will be used as our keynote reference in the accomplishment of our methodology. The steps in Zhang et al.'s methods include the following. Number 1. Literature search and compound selection. Zhang et al. Compiled a list of compounds that had antiviral properties against SARS and MERS coronavirus by using the query coronavirus and inhibitor and SARS or MERS or SARS-CoV or MERS-CoV in PubMed. Reading about the compiled compounds was done before the comparison with the three databases, namely Traditional Chinese Medicine Systems Pharmacology Database, the Encyclopedia of Traditional Chinese Medicine, and SIMMAP. Since our study varies from them in terms of the scope of compounds, for we examined plants locally available in Camarines Sur and Albay, whereas Zhang et al. Investigated Chinese compounds, we will be using a different set of databases. We will be using UPM's Philippine Traditional Knowledge Digital Library on Health, which includes a compilation of plants and natural products from documented DKDL researches conducted in the various regions in the Philippines, including Camarines Sur and Albay. We will also be cross-referencing with Dr. Jukes that facilitate in-depth plant, chemical, bioactivity, and ethnobotany searches using scientific or common names and other established related literature.
The second step is ADME screening of natural compounds. According to Zhang et al. 2020, oral consumption of Chinese herbal treatments after boiling in water is practiced by Chinese folk. Hence, compounds listed in the first part of the methodology will undergo an in silico integrative model of absorption, distribution, metabolism, and excretion to evaluate compounds that may be bioactive by oral administration. The indices used for the screening include evaluation of oral bioavailability, CACO2 permeability, drug-like value, and drug half-life. The threshold values indicating effectiveness for these four indices were more than 30%, more than 0.4, more than 0.18, and more than 3 hecto respectively, as recommended by Hu et al., a reference of Zhang et al. The values of these four indices were obtained from the TCMSP database. Oral consumption of herbal treatments is also observed in the Philippine local setting. We will be using the same threshold values for the conduct of our ADME screening. But we will also be using another database for the thresholds, namely ADMET-Lab, which provides a user-friendly, freely available web interface for systematic ADMET evaluation of chemical compounds.
Protein molecular docking. Zhang et al. 2020 also performed protein compound docking analysis in Autodoc 4. Their process of molecular docking will be followed by the researchers.
Molecules chosen from the previous steps were used as input for the TCM-SP, ETCM, and SIMMAP to search for plants containing that input. The plants were filtered by the numbers of antiviral compounds they contain. Those containing 2 or more antiviral compounds were selected for the next step. In comparison with the Zhang et al. Study, we will be using the mentioned databases for cross-referencing, namely UPM-TK-DLH, and established literature indicated in our literature search to select our compound.